So the continuation portion of this polarized light, basic properties, that means the wave model of light describes the light waves vibrating at right angles to the direction of propagation, with all vibration directions being equally probable. And this is referred to as common or non-polarized white light. Whereas in plain polarized light, there is only one vibration direction, no more than one vibration direction. Then polarized light is most commonly produced by the absorption of light having a specific vibration directions in a dichroic medium. Certain natural minerals such as tourmaline possess this property. But synthetic films invented by Dr. Edwin H. Land in 1932 soon overtook all other materials as the medium of choice for the production of plain polarized light. Any device which is capable of selecting plain polarized light from the natural white light, it is now referred to as what? Polar or polarizer. And the name was first introduced in 1948 by A.F. Halimond. Today, these polarizers are widely used in LCD, sunglass, photography, microscopy, etc. Then, careful specimen preparation is essential for obtaining good results in polarized light microscopy. The method chosen will depend on the type of material studied. In geological applications, the standard thickness for the rock thin sections is 25 to 30 micrometers. So in between, the thickness should be in between 25 to 30 micrometers. And also the specimen can be ground down with diamond impregnated wheels and then hand finished to the correct thickness using abrasive powders of successively decreasing grit size. Polarizing microscopes. The final specimen should have a cover glass which is cemented with optically transparent adhesive. The softer materials can be prepared in a manner which is similar to biological samples using a microtome. The slices between 1 and 40 micrometers thick are used for transmitted light observations. These should be strain-free and free from any knife marks. Next is about the parts. Actually, this polarizing microscope has two polarizing filters, which are termed as polarizer and analyzer. The polarizer is positioned beneath the specimen stage, usually within its vibration azimuth, fixed in the left to right or east-west directions, although most of these elements can be rotated through 360 degrees. Analyzer usually aligned with a vibration direction, oriented north-south, and it can move in and out of the light path as required. Then specialized stage, a 360 degree circular rotating specimen stage. It is to facilitate orientation studies with centration of objectives and stage with microscope. The next is about strain-free objective. The stress introduced into the glass of the objective during assembly can produce spurious optical effects. So in this type of microscope, objectives designed for polarized light observation are used, and they are distinguished from ordinary objectives with the inscription PPO and Pol on the barrel. Next is about centerable revolving nosepiece. Because the objective optical axis position, it may vary from one assembly to another. Many polarized light microscopes are equipped with this specialized nosepiece, and it contains a centering mechanism for individual objectives. Then strain-free condenser. The condensers, which are designed for the polarized light microscopy, have several features in common, including the use of strain-free lenses. Then eyepiece. About eyepiece, polarized light microscope eyepiece are fitted with a crosswire reticle to mark the center of the field of view. Orientation of the eyepiece with respect to the polarizer and analyzer, it is guaranteed by a point pin that slides into the observation tube sleeve. The next is Bertrand lens, a specialized lens which is mounted in an intermediate tube or within the observation tubes. A Bertrand lens projects an interference pattern. So these are all about polarizing microscopy. Next session we can discuss with electron microscopy. Thank you all.